Hello, my name is Tessa Behnke, and I'm a PhD student at the University of Nevada, Reno. In this presentation, I'm going to tell you a little bit about some of my dissertation work on greater sage-grouse. First, I'd like to start by thanking the people at the Applegate Field Office of the BLM and the Greater Hart Sheldon Conservation Fund, which funded this specific part of my research. Sage-grouse are a lekking species, which means that males congregate seasonally on areas called leks, where they display for females and try to get as many matings as possible. Here we have 3 males hanging out on a lek, and they're ready to use both visual and auditory cues to attract females. From many years of observations of these birds, we know that there's often a few dominant males or even a single dominant male called a master cock, which obtain a majority of these matings. However, we also know that not all matings are observable, even with careful observation, and other males may be getting additional matings off of the lek area. Females, as pictured here, are the choosy sex in this species. This is a female attending a lek, and as males compete for her attention, she has to assess their quality and choose which one to mate with. However, some of our previous research has documented hens mating with multiple males before laying their first clutch. Hens are trying to maximize their fitness and that of their offspring, but they don't have perfect information. Some hens may be hedging their bets on male quality or genetic compatibility with that male by mating with more than one male. Copulations in sage-grouse are very brief, and then the rest of the reproduction process is up to the hens. The males are no longer involved in raising the young or care of the nest. Sage-grouse females typically nest under sagebrush, although they're on their nest continuously once they lay the final egg. They do have to get up to take short breaks. This leaves an opportunity for another female to potentially come in and lay an egg in that female's nest. And a few cases of this have been documented in sage-grouse. It is very common in some other types of birds, particularly waterfowl. It's possible that this strategy could be related to kin selection, and females could be adding eggs to nests of their close relatives, which leaves then that hen to raise additional young that are still somewhat related to her at least. For my research, we're looking at nest parentage, and we want to know how often are hens mating with multiple males to sire their clutch, and is this a common strategy? We also want to see if any of the eggs within a female's clutch could be from another hen entirely, meaning that another hen has left a parasitized egg in her nest. Eventually, if a hen is successful, her nest will hatch, and then her and her precocial chicks will leave the nest for good, which gives us an opportunity to then sample these nests without disturbing the hen or the brood. We collected samples of nests of our known hens from our greater study in Northern Nevada. And with these nests, the membranes of these eggs are vascularized, which means there's blood flowing through them while that egg is developing. So they actually contain DNA from the chick. So we also collected whole eggs if there were any remaining in the nest after the rest of the nest hatched. And when we initially captured our females and marked them with a radio collar, we collected a feather sample and we used that as our maternal DNA sample. The only thing we don't have is DNA samples for our potential fathers, but I'll talk later about how we deal with this. All the molecular work for this part of the study was done at the USGS Molecular Ecology Lab in Colorado by Dr. Sarah Euler-McCants and Jennifer Fike. So shout out to them for excellent work. For this analysis, we chose to go with microsatellite loci and markers, which are these small repeats of sequences of DNA in the genome, and they're really variable. So there have already been 15 of these identified specifically for sage-grouse, and they're chosen because they do have a lot of variation and can be used to identify individuals. So we used these loci plus one for sex determination so that we would know the sex of each of these egg samples as well. Once the DNA is extracted from the eggs or feathers, we target those 15 loci with tags, and we sequence them for each individual. This is just an example graph of what that data might look like. So here we have colored regions for the different loci, and then the peaks within those regions are alleles. And so we score the alleles at those loci, and the combination of those is the genotype for the individual. For this analysis, we had really good quality reads and only had to exclude a few samples from our analysis. What I've done so far is using program Cervus, which we can do allele frequency, and parentage simulation, which gives us our expected ability to identify parents. And then I did a maternity check. So I'm checking all of the genotypes of the clutches against the putative mother sample to see if we have any mismatches there. We then used our clutches and our maternal samples to reconstruct our paternal genotypes since we didn't have the samples for those males. And we use these to see if there are any mismatches within the clutch to see if there could be multiple sires within that clutch. And for our preliminary results, I've checked about 250 of our egg samples so far, and I've identified 4 single locus mismatches. So there was only one mismatch for that individual. And there are no clear female offspring mismatches, so no evidence of nest parasitism. And our single locus mismatches could actually be genotyping error, or they could be a mutation in that individual offspring. What's really exciting is I have found 4 multi-locus mismatches, 4 eggs in 3 clutches, and I did that through the paternal genotype reconstruction. And so after accounting for our female genotype, there were still too many alleles left to have come from only one male, indicating multiple paternity in those particular clutches, which is really cool. So I believe we have found evidence for multiple paternity in our study area, but no evidence of nest parasitism by females. And our next step is to verify these results with some other programs that can run every possible combination of those alleles and figure out how many possible genotypes there are. We also need to assess our genotyping error rate, and we can do this because we sequenced a subset of our samples twice to make sure that we can have an error rate, and that might be the explanation for those single locus mismatches. We can tell a little bit more about that and whether or not those are actual genomic differences. We can also use our paternal reconstructed genotypes to look for those dominant males that I talked about earlier. So many of our clutches are taken from a small area, and so we can see if we can pick up a signal of either a single or a couple of males that are getting most of those matings. And with that, I would like to thank my numerous field technicians that helped with this work over the years, my program at UNR, and our funding agencies that funded the main part of this project over the last several years. And thank you to you for tuning in, and I'm happy to take any questions. My email is tbehnke@nevada.unr.edu. Thank you.